Hello, everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss the insects that independently invented agriculture. So let's jump right in. In the last video, we covered all the non-hexapod arthropods, the chelicerates, myriapods, and crustaceans. Today, we are moving on to the clade hexapoda, the six-legged arthropods. Crustaceans transitioned to living on land multiple times. One of the most well-known examples is the isopods, often called pillbugs or roly-polies. All isopods have gills, but one group of isopods, the suborder Onicidia, lives on land. They make up a whopping 82% of terrestrial crustacean biodiversity. Because they have gills and also lack a protective waxy epicuticle similar to what insects have, terrestrial isopods are confined to cool, damp places like under rotting vegetation. Based on their paleogeographic distribution, this clade appears to have made landfall between 350 and 300 million years ago during the Carboniferous, while their earliest fossils show up around 100 million years ago in the late Cretaceous. Semi-terrestrial amphipods of the superfamily Talatroidea originated 140 to 138 million years ago in the early Cretaceous. One terrestrial amphipod, Talatroides topitotum, has earned the common name the cosmopolitan landhopper because it has an almost worldwide distribution. Decapods, the crabs, lobsters, and shrimp, may have terrestrialized a bit earlier in the lower Jurassic. The crab Poteminotes loveni from Kenya and Uganda are found in terrestrial habitats up to 3,060 meters above sea level, and the crab Geothelphusa haetuan is known from cloud forests growing at approximately 2,000 meters above sea level in the mountains of Taiwan. The heaviest terrestrial crustacean is a decapod, the coconut crab Burgess latro, which is actually a hermit crab, a member of the hermit crab superfamily Pageroidea. The coconut crab belongs more specifically within the family of terrestrial hermit crabs Cenobidae, alongside its closest living relatives, the 17 species of the genus Cenobita. While the coconut crab is often not associated with hermit crabs, juvenile hermit crabs do use empty gastropod shells to protect their abdomen, and adolescents sometimes use broken coconut shells instead. Furthermore, despite their names, neither the coconut crab nor all the other hermit crabs are in fact true crabs. True crabs are members of the decapod clade Brachyura, while hermit crabs are part of Anomura, the sister clade to the true crabs. The hermit crabs are far from the only examples of decapods that are called crabs while not actually being true crabs. For example, porcelain crabs are more closely related to squat lobsters than they are to true crabs. Squat lobsters aren't true lobsters either, but let's stick with crabs for now. Another example of false crabs are the king crabs, which are also large hermit crabs, but they are not closely related to the terrestrial hermit crabs like the coconut crab. King crabs are more closely related to the fully marine hermit crab family Paguridae. So hermit crabs evolved giant sizes twice, once in the sea and once on land. This convergent evolution of crabs happened so many times that biologist Lancelot Alexander Borradaile coined the term carcinization to refer to, quote, the many attempts of nature to evolve a crab, close quote. The decapod with the most extreme adaptations to terrestrial environments is the Jamaican bromeliad crab Metopoleus depressus. For most crabs, parental investment stops once the larvae detach from the mother's abdomen. In aquatic environments, decapod larvae are planktonic, but terrestrial or semi-terrestrial decapods have shortened the developmental process. The larvae are direct developing from eggs and remain adhered to the mother for a few days. Indeed, parental investment has evolved in a few lineages of terrestrial crabs, like Cesarma jarvisi, who inhabits large snail shells with her offspring. However, the Jamaican bromeliad crab puts them all to shame. The mother Jamaican bromeliad crab lives with all her offspring in a large, wait for it, bromeliad, which offers safety and provides food. Fascinatingly, the mother crab may place snail shells in the water where her eggs are laid for extra calcium carbonate supplementation. There can be multiple overlapping generations, up to three, that all inhabit the same bromeliad, but interestingly, there is only one reproductive female in the colony. Certainly, other females live in the colony, but they participate in defense and care for the younger generations. This is an example of eusociality in a crab. Eusociality has also evolved in the sponge-dwelling shrimp, genus Synalpheus. This genus shows the whole range of sociality from asocial pair bonding to true eusociality. Outside of crustaceans, eusociality has evolved in the naked and demaraland mole rats, and most famously, bees, ants, and termites are eusocial insects. We'll come back to them. Moving on to microcrustaceans, ostracods like Mesocyprus, copepods like Epactophanes, and fairy shrimp like Bryospilus are associated with water films on surfaces such as mosses, epiphytes, and soil in humid forests. One ecological route by which crustaceans became adapted to terrestrial environments may have been to transition from saltwater to moist sand or soil before switching to land. Recent phylogenies have placed the crustacean clade Remipedia as sister to Hexapoda. Remipedes are small, blind animals with many repeating segments found in saline aquifers. The clade Cephalocurida, also known as the horseshoe shrimp, may be sister to the Remipedes, but more genetic data is needed to confirm this relationship. Both Remipedia and Cephalocurida are fully aquatic groups, so the rise to a terrestrial existence for hexapods must have occurred after their common ancestor with these clades. So, what morphological adaptations did hexapod ancestors develop to live on land? One of the most obvious is their waxy epicuticle that prevents water loss. Another is the internalization of all their respiratory structures. Tiny crustaceans, like copepods, have no need for a respiratory system because they can simply respire across the body wall. However, larger crustaceans need gills or other respiratory structures. Before we get into this, we first need to understand that an arthropod's body is divided into segments. In some groups, like myriapods, there are many repeating segments, but in other groups, like spiders, many of the segments have been fused. In crustaceans and hexapods, the animal is divided into a head, thorax, and abdomen, where the abdomen is sometimes referred to as the tail. Along these segments are appendages used for a wide variety of functions. Let's take the lobster as an example. On the head are antennae and antennules that function in sensing the environment. The mandibles are the jaws. Behind the mandibles are two or more pairs of maxillae that sweep food into the mouth. In some crustaceans like lobsters, there is an additional pair of appendages behind the maxillae called maxillipeds that also help move food into the mouth. The chelipeds are the large claws. Then there are the thoracic limbs. On the abdomen are the pleopods. And finally, the last segment of the abdomen possesses fan-like uropods. Crustaceans are a veritable Swiss Army knife of appendages. Arthropod appendages can then be divided into separate branches, such as the epipod, exopod, and endopod, and any or all of these can bear gills, allowing mechanical aeration. Some crustaceans also have internal gills. A crawfish has holes on the base of its carapace that allow oxygenated water to be pumped into its branchial chambers, which contain the gills. The internal gills are also extensions of the limbs, specifically the maxillipeds and walking limbs. The first gill is a podobranch, the second and eighth gills are arthrobranches, and gills 3 through 7 are pleurobranches. Returning to the isopods, aquatic isopods have gills on their pleopods, but in terrestrial isopods, the pleopod gills are infolded, forming what is called the pleopod lung or pseudotrachea. As for the hexapods, they don't have gills, with the exception of some larvae. Instead, insects have a tracheal respiratory system, which is a series of ectodermally derived chitinous branching tubes connected to holes along the body called spiracles. Note that these spiracles are totally unrelated to the spiracles we discussed in the lamprey's tail. Trachea have independently evolved in multiple lineages of arthropods, such as myriapods, opiliones, the daddy longlegs, Raciniulae, the hooded tick spiders, Solifugae, the camel spiders, and some true spiders. Multiple ectodermal tubes are just one way to solve internal ventilation. Arthropods have an open circulatory system where their organs are bathed in blood, which is quite different from our closed circulatory system where our blood is contained in veins and arteries. Arthropods also have bluish rather than red blood because they use hemocyanin for oxygen transport, which is copper based, unlike our iron based hemoglobin. When the colorless copper 1 is oxygenated, it becomes copper 2, which is blue. So, similar to how osteichthyans evolve lungs to increase the surface area for oxygen intake as they evolve larger sizes, arthropods too develop more surface area for oxygen intake, just along their body wall instead. And just like lungs, hexapod trachea have undergone numerous modifications, both for sound reception and production. Regarding sound reception, hexapods don't have ears like we do. They have a thin tympanal membrane backed by a tracheal sac that is associated with sensory neurons. And there usually aren't just a pair of them on the head, they may be found all across the bodies of hexapods. For instance, the legs of katydids, the abdomen, thorax, and even wings of Lepidoptera, and the thorax of mantises. As for sound production, cicadas use an organ called a timbal, which is also a thin membrane backed by a tracheal sac. However, instead of just passively receiving sound, the membrane itself vibrates and the sound is amplified by the tracheal sac. So now let's dissect the diversity of hexapods. Hexapoda splits into the clades Archaeognatha and Dicondylia. Archaeognatha contains Columbella, which are the springtails, and Protura, the coneheads. Formerly Columbella, Protura, and the third non-insect hexapod order Dipleura comprise the clade Antagnatha, which means inside mouth, because their mouth parts are internal, while all insects are Ectognathous, which means outside mouth, because their mouth parts are external. As with so many other phylogenetic relationships we've discussed throughout the series, this topology turned out to be a bit incorrect. Dipleurans are actually more closely related to insects than to columbellans and proturans. The morphology of columbellans, proturans, and dipleurans is pretty similar. All are small, wingless, multi-segmented arthropods with sensory filaments on the tail called cerci. Unsurprisingly, they have similar ecology too. They consume decaying organic matter, contributing to the return of nutrients to soil. The earliest hexapod fossil is the Columbellan Riniella from 410 million years ago. This fossil hails from a Scottish geological formation known as the Rhynie Chert. This formation dates to 411 to 407 million years ago, which is the early Devonian period and appears to preserve a very early terrestrial community. According to one paper, quote, these translucent rocks were formed in a hydrothermal hot spring setting in which silica-rich water inundated and silicified nearby vegetation, including a number of the animals, all arthropods, creating a near-perfect record of this early terrestrial ecosystem, close quote. The community contains charophyte algae like Paleonatella, early vascular plants like Aglaophyton and Asteroxylon, fungi of various clades, and, as said earlier, arthropods. The arthropod fauna includes the fairy shrimp Lepidocaris, millipedes and centipedes, the opilione, Eophalangium, mites, and trigonotarbids. Trigonotarbids are an extinct clade of arachnids that look similar to spiders but with some important differences. For instance, trigonotarbids can't spin silk and they have a segmented posterior region. In 2009, a popular UK newspaper covered some then recent work by paleontologist Russell Garwood on trigonotarbids, and some online comments on this article were made by young Earth creationists. One comment in particular said, quote, they look just like spiders do today, 300 million years and no change. Kind of hard on the old evolution theory, isn't it? Close quote. Of course, trigonotarbids aren't modern spiders and lack many of their diagnostic characteristics. This type of argument is what we've termed the Haran-Yahya tactic, and we've done a whole video on it. As paleontologists Garwood and Edgecombe insightfully note, quote, this altercation encapsulates perfectly a common creationist mindset, one based on cursory and often incorrect, observations coupled with complete ignorance, or at best patchy understanding, of the scientific context framing an argument. If they succeeded, this would stifle the colorful world of scientific discovery and reduce it to a Manichaean palette of unquestioning certainty and unknowable unknowns. Close quote. The Rhynie chert clearly contains non insect hexapods, but does it contain insects too? At present, that is unclear. The well known fossil mandibles Rhyniegnatha appear to more likely belong to a myriapod than an insect, and there is debate over the affinities of Leverholmia. It may be a hexapod, possibly even an insect, or some other kind of mandibulate. This is because the fossil lacks a head. Though molecular clocks routinely give dates for the origin of insects ranging from 430 to 370 million years ago, the late Silurian to the late Devonian, the first definitively insect fossils date to 328 to 324 million years ago, the Middle Carboniferous. That's a gap of at least 42 to 102 million years. So, is there really a gap in the fossil record here, or do molecular clocks need to be recalibrated for this clade? To answer this question, we need to go back to the Carboniferous. Remember from the lava lizard's tale that this was a period of great diversification for early amniotes. From the first records of terrestrial arthropods in the Silurian to about 331 million years ago in the Carboniferous, there are no definitive insects, but non insect hexapods, myriapods, and arachnids are known. Then, about 328 million years ago, the first insects appeared and radiated extensively. Interestingly, the stratigraphic appearance of fossil insects matches their phylogenetic relationships quite well. For example, wingless hexapods show up well before insects do. Paleoptera, which includes the dragonflies, damselflies, and mayflies, appeared before Polyneoptera, which includes earwigs, grasshoppers, stick insects, mantises, roaches, and termites. Polyneoptera appeared before Paraneoptera, which includes lice, true bugs, and thrips. All these groups appeared before Holometabula, which contains all the insects that go through larval, pupal, and adult stages of development. And stem Holometabula appeared before crown Holometabula. This is exactly what we expect on evolution. It is worth pointing out that there are over 5,500 described occurrences of insects from the late Carboniferous through the Permian fossil record, but there are fewer than 20 described occurrences of non insect hexapods. Clearly, there is a bias in the fossil record, so we should tentatively take the lack of insects in pre Middle Carboniferous rock as evidence of their absence. Of course, one of the main reasons for the rapid diversification and spread of insects was their wings. Debate has raged for decades over whether wings arose from elements of the legs or thorax, and recent developmental, genetic, and fossil data has been marshaled in support of one particular hypothesis of wing origin. However, to understand it, we first need to talk about a totally extinct clade of early insects called Paleodictyopteroidea. A mouthful. These insects are one of the first groups of insects to appear in the fossil record, and they were the only major group of insects to be knocked out by the Permian Triassic mass extinction. They are considered either sister to dragonflies and mayflies or sister to all other insects. Their primitive characters make them hard to place. Similar to modern aphids and stink bugs, Paleodictyopterans have beaks for piercing plants to suck fluids, and they range in size from a third of an inch to almost 2 feet. Unlike all other known insects, they had 3 pairs of wings, not 2, with the third pair being small winglets on the frontmost thoracic segment that probably functioned in flight stabilization. What's most intriguing about Paleodictyopterans, though, is their wing pads. Wing pads are the underdeveloped, immobile, flattened structures on insect larvae that will become wings, and in this group, the wing pads are attached not just to the thorax, but also to limb tissue. This is important because developmental studies have indicated genes and tissue from the thorax and limbs are involved in wing generation. In other words, genes and tissue were co-opted from the thorax and limbs to form the wings. Wings didn't spring up from nowhere. But once insects alighted on their wings, they took to all regions of the globe, surpassing all other known clades in terms of species. As entomologist Robert M. May said, Quote, indeed, to a good approximation, all species are insects, close quote. One estimate pins insects as containing more than 1 million species. 30 to 40% of those are just beetles. Famously, American entomologist Terry Irwin counted just beetles on the Panamanian tree Luhea seemanii, yielding 1,100 species. Following that, there are around 160,000 species of butterflies and moths, 160,000 species of flies, 120,000 species of bees, wasps, and ants, and 100,000 species of true bugs. With the exception of the true bugs, all those groups are found within Holometabula, the metamorphosing insects. Remember from the previous episode that all ecdysozoans grow in discrete stages separated by molting. For some organisms, each stage, called an instar, is identical in shape but not in size. In other words, the juvenile looks like a miniature adult. This condition is called being ametabolous, and it is found in the non-insect hexapods. Then, as wings were evolving in stem insects, they had to deal with these while molting too. One option would be to make small articulated winglets that grew in size with each molt. However, insects went with a different option. They developed immobile wing pads that morph into articulate wings during the adult molt. Another change that occurred was the adult molt became the final molt. As one paper points out, quote, the cessation of adult molting was likely related to making the wing as light as possible. Wing reduction occurs by the death of the epidermal cells that secrete the cuticle of the wing membrane after adult emergence. However, without these cells, the cuticle of the wing membrane cannot be made again. Close quote. The only extant insects that molt their wings are Ephemeroptera, the mayflies, one of the two most basally derived clades. They have a subadult instar with wings, but the wings are thick, making this instar a very awkward flyer. Interestingly, even in insect clades that lost wings like fleas and lice, they never regain the ability to molt as an adult. Thus, the loss of adult molting appears to be irreversible for insects. This condition of dividing life stages into a nymph without wings before molting and an adult is called being hemimetabolous. This characterizes all insects that are not members of the clade Holometabula. For most hemimetabolous insects, the nymph and the adult occupy similar habitats. After all, they tend to be rather similar in body shape, except for the wings, of course. The evolution of this condition involved primarily post-embryonic changes, but the evolution of the holometabolous condition involved embryonic changes too. As mentioned earlier, being holometabolous involves having an egg stage, larva, pupa, and then adult. The importance of this is that the larva and adult are developmentally uncoupled. The larva can have reduced motility but an enhanced capacity for food exploitation, while the adult can have its own adaptations for dispersal and reproduction. It makes absolute sense then why insects took over the planet. As a side note, thrips and some hemipterans like scale insects and mealybugs independently evolved holometabolism. To recap, the ancestors of hexapods were ametabolous, the hemimetabolous condition evolved in stem insects, and finally, the holometabolous condition evolved in stem holometabola. We can therefore broadly see the stepwise acquisition of metamorphosis in living hexapods. Insect metamorphosis is controlled by hormones called ecdysteroids. Similar to those thyroid hormones we met in the axolotl's tale, ecdysteroids bind to a class of animal-specific proteins called nuclear receptors, which themselves bind to DNA and regulate it when activated. The insect's brain secretes the hormones prothoracicotropic hormone and insect insulins, and these cause the prothoracic glands to secrete the ecdysteroid known as ecdysone. Ecdysone then undergoes modification to become 20-hydroxyecdysone, which is the active hormone. Production of this and other ecdysteroids is upregulated during molts. Unsurprisingly, the genes involved in holometabolous development long predate the insects that use them. Now that we are within holometabola, we can get to the clade Hymenoptera, the bees, wasps, and ants. Really, all hymenopterans are wasps. Bees and ants are just highly modified wasps. The earliest fossils of hymenopterans come from the 237 million year old Madagen Formation in Kyrgyzstan, which is the middle to late Triassic, such as Xylinus, Triasoxyella, and Microxyella. The earliest bee and ant fossils, however, only date to the late Cretaceous. Sphecomirma is one of the earliest ants, dating from 92 to 79 million years ago, and was specifically predicted by paleontologists based on the diversity of ants in the Eocene. Just this year, a team of researchers published a paper documenting antennal sensilla for alarm pheromone detection and nestmate recognition in Gerontoformica ants, found in 100 million year old amber, indicating that even early ants possessed advanced sociality. Just like the Jamaican bromeliad crab we met earlier, ants and many bees possess eusociality. One of the early attempts to understand how hymenopterans developed eusociality was done by British evolutionary biologist William D. Hamilton. He posited that hymenopterans are eusocial because of their odd genetic system called haplodiploidy. Males are the result of unfertilized eggs, which are haploid, or containing only one set of chromosomes, while females are the result of fertilized eggs, which are diploid, or having two sets of chromosomes. As a result, sister ants are more closely related to each other than these sisters are to their own daughters. It works like this: an individual female ant contains half of her mother's DNA, but all of her father's DNA because he only has one set of chromosomes to give. If half of her mother's DNA is half of her DNA, then we can represent that as 1/2 times 1/2, which equals 1/4. However, 100% of her dad's DNA is the other half, which we can represent as 1/2 times 1, which equals 1/2. Now we add 1/4 plus 1/2 to get 3/4. Thus, each full sister, meaning sisters resulting from the same parents, share 3/4 of their DNA with each other, while they only share half of their DNA with their own offspring. Due to the genetic closeness between sisters, Hamilton reasoned, ants should therefore put in more effort to raising more sisters than to raising offspring. This hypothesis seemed plausible because all known eusocial animals, except termites, were hymenopterans and thus haplodiploid. One issue with this hypothesis arose when more eusocial species were discovered that lacked the haplodiploid system. Naked mole rats, Synalpheus shrimp, ambrosia beetles, and the Jamaican bromeliad crab are all traditional, God-fearing diploids. However, there is still an important difference between hymenopterans and those other groups. The true diploids retain the ability to become reproductively active under certain conditions, while hymenopteran workers can never mate. Another issue is that mothers are more closely related to their sons, sharing half their DNA, than brothers are to each other, sharing only 1/4 their DNA. Remember, males lack paternal DNA. Therefore, there have to be more females than males in a nest for haplodiploidy to favor worker behavior. As it happens, hymenopterans are indeed capable of changing the sex ratios of their broods. Many solitary insects have two broods per year, a condition called bivoltinism, and with that as the starting point, biasing the sex ratios of those broods would have important effects for raising offspring. For example, if a parental pair has their first brood in the spring, then any females produced in that brood would help raise their sisters produced in the second brood. What would happen then if that newly evolved helping behavior in sisters caused a skew in the sex ratio of broods? That can be accounted for by mathematical models encompassed under sex allocation theory, which we discussed briefly in the seal's tale. Remember from that video that there isn't always a 1-to-1 ratio in terms of resource allocation for sons versus daughters. Producing a daughter guarantees mating, but with few offspring. However, producing a son doesn't guarantee a mating, but with the potential to produce very many offspring. In the case of our hymenopterans, once producing non-mating female workers who aid the mother evolves, the first brood becomes essentially all female workers, while the following brood contains roughly half males and females. After all, you want individuals who help you out to be born first, so you don't have to do the job of raising the generation alone. So the origin of eusociality isn't just a matter of haplodiploidy, but also involves lifetime monogamy to ensure relatedness among offspring, a bivoltine life cycle where females overwinter, and flexibility in the evolution of sex ratios. Possessing a combination of these characteristics allowed for the evolution of eusociality in hymenopterans. With all of that covered, let's now get to the leafcutter's tale. Leafcutter ants are split into two sister genera, Atta and Acromyrmex, that contain a total of 47 species which are native to South, Central, and Southern North America. Broadly, ants are divided within a colony into specific jobs: the queen, soldiers, and workers of various castes. As you have no doubt seen on a sidewalk or maybe even in your house at some point, some ants find food and bring it back to the nest. Unusually, leafcutter ants of the Mediae caste don't snip leaves to feed the colony. Instead, they bring the leaves back to the nest and have workers of another caste, called minims, mash up the leaves to be put in compost piles. These compost piles feed fungi, which are always members of the family Agaricaceae, and the ants feed the fungi to the colony. Yes, these ants are practicing agriculture, a feat we haven't seen since the farmer's tale. And like human farmers, pests attempt to take over their gardens. The Ascomycete, Escovopsis, is only known from ant fungus gardens. However, also like human farmers, fungus growing ants use pesticides. The filamentous bacterium, Pseudonocardia, lives on the ants and produces antifungal compounds to limit the pest's growth. But these two genera are not the only fungus growers. All genera within the taxonomic tribe Attini are obligate fungus growers. They are all New World, with species stretching from the northern US to southern South America. Originally, fungus growing ants would have just gathered fungi as part of their foraging, but specializing on fungi specifically may have helped early fungus growing ants avoid competition with other ants. Entomologists have divided the agricultural techniques of fungus growing ants into multiple types. The first type is called lower agriculture, which involves cultivating a wide range of species within Agaricaceae. The second type is called coral fungus agriculture, which cultivates fungi of the family Pterulaceae. This is mostly done by members of the genus Aterostigma. The third type is called yeast agriculture, which is mostly done by Sipho-Myrmex. The fourth type is called generalized higher agriculture, which is practiced by three genera of non-leafcutter ants. Finally, leafcutter agriculture is the type mentioned previously. When a phylogeny is constructed for fungus-growing ants, the leafcutter ants nest within a larger clade that practices generalized higher agriculture, and that clade is nested within a larger clade that practices lower agriculture. And the queen ant even takes a small sample of the fungus with her when starting a new nest. Another form of agriculture by some ants is tending to aphids. Similar to how humans milk cattle, some species of ants massage aphids into producing a sweet substance called honeydew. When aphids pierce the phloem of a tree, the sugary liquid is not fully digested, and ants can coax aphids to excrete some of the liquid by gentle taps on the rear. I have heard the claim that there is at least one species of aphid whose rear looks like the face of an ant, which allegedly entices the ant to milk the aphid. At the time of writing, however, I have found no evidence to corroborate this claim. Link a citation in the comments if you can find this species. Interestingly, honeydew may be the manna described in Exodus that the Israelites consumed in the desert. And associating with ants has been shown to increase the populations of aphids, just like how humans have increased the populations of livestock. In one instance, a species of aphid has flipped the script on ants. Paracletus simiciformis tricks the ant Tetramorium into letting the aphid into the ant's colony. Once inside, the aphid drinks fluid from ant larvae. Finally, ants are not the only animals to have independently invented agriculture. Ambrosia beetles and termites also chew up plant material and deposit it into compost piles to grow fungi that they then eat. The moral of this tale is that humans are not unique in developing agriculture. Insects did it long before we did, and they've been doing it very efficiently ever since. So, thanks for watching, and we'll see you all next time.